So interphase was last week. Now we're going to look at the other stages, prophase, metaphase, anaphase, and telophase, and what goes on during those phases. So interphase, where the chromosomes are loosened to allow transcription and DNA replication, and interphase was the cell being a cell. So then we have prophase. I don't think I mentioned last week about the way I remember the stages of mitosis and the cell cycle in general. It does not want to come off. But you can remember it as I, P, MAT, C. So interphase, prophase, metaphase, anaphase, telophase, cytokinesis. So those are the different steps in the process, I, P, MAT, C. This is the entire cell cycle. So if you go from interphase to cytokinesis, that's the cell cycle. But if you just go prophase to telophase, that's mitosis. So interphase to cytokinesis is the whole cell cycle, and then it will repeat itself and it goes in a circle. But prophase to telophase is mitosis. Now mitosis is just the division of the genetic material and the nucleus itself. So that's what is the difference between the two. The cell cycle is the division of everything, but the mitosis is just the division of the nucleus and the genetic material. So when we start with prophase, now we're dividing the genetic material and the nucleus. The other thing to really keep in mind with this, even though we learn it as prophase, metaphase, anaphase, telophase, and very distinct stages like that, it's actually a smooth, continuous process. So when we look at them under the microscope, sometimes you're going to see some that are kind of like in between stages. And it'll be, well, is that one prophase, or is it actually, getting into metaphase? Because it's one smooth, continuous process. It might look like in between stages sometimes. So that's why we have something called early prophase, and you might have something that's like pre-metaphase or something like that, because it's a smooth, continuous process. So early prophase is when the chromosomes kind of start to condense and you can start to see them. Also, the nuclear envelope is beginning to break down. The chromosomes are beginning to pack tightly together, and they're really starting to be visible for the first time.
Guess what I figured out?
What did you figure out?
So for Spanish, I have to write a script, and I was getting really annoyed of having to press shift and type. So I figured out how to put caps lock on my Chromebook.
That's very exciting.
I didn't know you could do that, but I just looked it up and then it worked.
It's good when you learn technology better. So when we get to your test, we're not quite there yet, because we still need to learn meiosis. But when we get to your test, you will have to identify the stages of mitosis, the pictures of them. So make sure that you are familiar with the different stages. All right, so interphase, of course, is a normal cell, normal appearance. You cannot see the chromosomes yet because they have not condensed, and the nucleus is usually uniform in color. It looks the same throughout. But when we start to get to prophase, the chromosomes are starting to be, you can see the chromosomes. They look like darker lines inside the nucleus. The centrosomes move to the opposite ends of the cell. The centrosomes and centrioles are kind of the same thing. So they're moving to opposite ends of the cell because they're going to start to grow what are called spindle fibers. And and these spindle fibers are going to help attach to the chromosomes and other organelles, and they help to make the cell lengthen kind of oval shaped in in animal cells. Plant cells, they don't get oval shaped because they have a cell wall, but they start to get oval shaped and expand the cell, getting ready for it to separate. The nuclear envelope is also going to start to break up, so it starts to dissolve so that the division can really start to take place. So this is all happening in prophase. I see you have an Onyx Storm book there.
I do too.
I have read the other books. I'm in the middle of another series though. It's like, do I stop the other series to read this book? I don't know, you have to tell me if it's good.
I'm so excited for the new Hunger Games book.
Yes.
There's a new Hunger Games book?
Yes, it's during Hamish's games, and then there was gonna be a movie about it. They already had the movie release date out.
It better be.
Hamish's game. Oh, like when he was in it.
Yeah, he did it.
Oh.
So I'm really excited for that.
That's gonna be so good. It better be.
It
Read the book's good. I've never read the book.
The Hunger Games books? Yeah,
They're good.
they're really
You should
good.
do that.
I'm surprised you haven't read them.
You
Have you watched the movies?
Sometimes if I watched all the Harry Potter movies, and then my mom decided to tell me that she had the books, and I just couldn't get into the books.
I think they're just wrong books.
Books are always better than the movies.
It'd be so
Like if I watch the book and then the movie, then I can read it. Like The Boy in the Striped Pajamas, I love that book and the movie, but like other movies, I just can't get into.
Nah, books are always better.
Who? The books are always better than movies.
Always.
I've only read the books. I haven't seen movies.
Always better.
The Hunger Games movies, you haven't seen them?
The movies are good too. In fiction and film, I'm pretty sure you read the Hunger Games and watch the movie.
Yeah.
They're really good.
Oh, something else I read the book was not the movie. I didn't watch the new movie because I liked the book.
The Dune? I've only watched the movies. I don't read the book.
Alright, early and late prophase. So here's what it looks like in early prophase, where the chromosomes are condensed inside the nucleus, and it really does look, I mean, obviously not orange, but they're usually a purpley color because the cells have been stained. That's why they're purple colored. In fact, well, when we stained our cheek cells, we used kind of a greenish, bluish color. But anyways, they have a dark lines within the nucleus, and that's the chromosomes that are condensing. Later prophase, they're starting to kind of line up toward the middle, and the nuclear membrane is going away. So this is in Google Classroom. But late prophase, the membrane has dissolved almost all the way of the nucleus, not the cell. The nuclear membrane has dissolved. Because remember, mitosis is nucleus and genetic division. It is not cell division. Mitosis is nuclear and genetic division. The spindle apparatus is forming, so all of these little fibers here, these are the spindles, and you can actually see these under the microscope as well. And these are your centrioles that are forming at either end of the cell. They help line everything up. All right, then you have metaphase. Metaphase middle, that's how I remember it. Metaphase middle. Everything gets aligned in the middle of the cell. Usually the chromosomes are in their most condensed form during metaphase, and everything basically just aligns in the middle. That's really all that happens during metaphase. Anaphase is pretty easy as well. Anaphase, they get pulled apart. Anaphase apart or away, bless you. So anaphase, everything's pulled apart. Metaphase middle, anaphase apart. And during anaphase, everything gets pulled toward opposite ends of the cell. So those two stages are pretty easy. Anaphase apart, metaphase middle.
I'll post it on my Instagram. I'll take pictures on my Instagram. Post it on the gram.
There's another fun thing this week with all the TikTok drama.
Oh, it being banned for 14 hours.
Serious health issues if done incorrectly or too frequently.
Don't crack your neck too
Wrist, stroke,
Oh.
artery tear,
I
osteoarthritis,
cracked my neck all the time.
neck and spine damage. I'm done.
Thank you. I already have spine damage, so okay, me too.
Alright, so metaphase, they're basically lining up in the middle. Metaphase, middle. Anaphase, they're being pulled apart. Now at this point, the sister chromatids have separated. In this point, they're all still attached together. So they're replicated and they're made a copy of themselves. The copies are still together. At this point, they are no longer copied together. One copy of the genetic information has gone this way and the other set has gone this way. So genetic information is now separated, and that's because each side is going to become a new cell. So now we've got enough information to be a cell over here, enough information to be a new cell over here. The cell information has been pulled apart. The final step of mitosis, because PMAT is the actual mitosis, the final step is telophase. The chromosomes arrive at the spindle poles. Telophase is kind of the opposite of prophase. In prophase, everything's condensing. In telophase, everything is unraveling or uncondensing. And also the nuclear membrane reforms. So prophase and telophase are the opposite of each other. So now we have the chromosomes unraveling and we have the nuclear membrane coming back.
Miss Ayers, would you consider scoliosis a mutation? Because I'm putting it down as a mutation.
Honestly, I don't know.
I mean, it's not normal.
Well, but I don't know if it's a mutation or if it's a
If it's like
genetic
scoliosis.
disease or genetic disease caused by mutation. I'm not sure.
Scoliosis is a complex condition that can be caused by genetic mutation, but it's not always genetic.
I'm putting that
80%
down.
of cases, the cause of scoliosis is unknown. Mine's because most of the
My aunt has
ones that I've
scoliosis.
heard of are because of gymnastics. If the ones I know.
My aunt has scoliosis.
Scoliosis. I don't think my parents have scoliosis though, so.
My
Mine's
aunt
not
has it, and I have it. My spine's curvy.
I found out I had scoliosis because my coach asked why I walked with one shoulder like down.
The British.
The British are coming.
I got a curve in my spine. That's why I can't do some workouts because I'm like, oh my god.
Mom doesn't hurt anymore. It used to be really bad.
Mine was bad in acro and it's still bad.
We went to the cadaver lab last year for anatomy, and the female cadaver that we were shown had severe scoliosis. So it was very interesting to see because when they showed her spine, you could see the curve. It was really-
I have an x-ray of mine, like you can see my curve in my spine on the x-ray.
Me too. My great grandma has like scoliosis. She has all the oses.
The
And she
osis.
literally has like 4 things on her
Mine's
back,
like-
and she has like a pump in her stomach of like morphine because of how bad it is.
It's terrible.
Oh, that's curvy.
All right, so this is showing telophase, not to be confused with cytokinesis, which is also occurring here. So telophase is the nuclear membrane coming back and the chromosomes decondensing and going back into their like stringy form. Then we also have cytokinesis, which is happening here with the cytoplasm, where the cytoplasm is pinching in. So 2 different things happening at the same time, and this is in Google Classroom. Okay. So here is showing the spindle fibers. The centromeres are the really bright areas, and then all the spindle fibers you can see extending out of that. This is in Google Classroom. It's a great summary. It shows both a plant and an animal. Here's our plant here. You can see the very distinctive cell wall surrounding it. and the animal one. Now, we are going to do a lab, I forget which day, we'll do a lab soon, where we look at slides under the microscope of plant and animal. We have a lot more slides of plant. I don't know why we have so many plant slides of mitosis, and we have very few animal slides of mitosis. So most of the ones we're going to look at in lab are actually plant ones, but the plant ones we have are very, very good. So you'll be able to see lots of plant cells going through mitosis. So the animal ones we have are not very good. I think there's only one or two really decent ones. But the animal ones we have are really good, so you'll be able to see the plant ones. The plant ones, you'll be able to see that pretty well. But this is in Google Classroom. It shows plant, animal, and then a drawing and also a description. So it does a great job of kind of giving you lots of options to break it down.
The middle picture reminds me of, on that side, the pink one reminds me of like a toilet bowl and like the cleaning powder, like the toilet bowls.
That is very descriptive.
Like the cleaning, I've seen videos where they just put a bunch of chemicals in toilets and clean them.
It's probably not the best. It's just
That's
mixing
how-
chemicals.
they are,
That's
and
how I feel every
It's
time I
like,
do.
I'm like, they're making mustard gas, so like, it reminds me of that.
Yeah.
It's like the pink.
Oh, when I was in the bath, I'd like put like 45 shampoos into a cup and like.
Yeah, I would make a potion.
Yeah.
Like, I've done chemicals in the toilet bowl every time. They put ice in there and
Just
everything.
airs for our test. Is it gonna be like an actual cell, or is it gonna be like the one that like on the side?
You will be expected to identify an actual cell.
Okay.
So I would get in the practice of identifying these pictures instead of these pictures. You will have a few of these because there will be questions I ask about, especially meiosis, about, stages of meiosis, because there's like things like meiosis 1, meiosis 2, prophase 1, prophase 2, and stuff like that. So there will be a few that all use pictures like this, but as far as identifying metaphase 1, metaphase, prophase, anaphase, and things like that, you will need to know these pictures. So make sure you're getting in the habit of looking at these. But these are metaphase, anaphase, and telophase. Alright, so cytokinesis. Cytokinesis is the division of the cytoplasm. This is not part of mitosis. Again, I stress that a lot. Cytokinesis is after mitosis when the cytoplasm is dividing, and it's different between plant and animal cells. It is a different process. Different between plants and animals. Why do you think that is? Why does the cytoplasm need to divide differently between plants and animals?
Because they're two different cells.
I mean, yeah, but what does a plant cell has that an animal cell doesn't?
Cell wall.
Cell wall. The cell wall makes it so that it's harder to divide the cytoplasm. With an animal cell, it's pretty easy to just kind of like boop, pinch it in half and you're done. But with a plant cell, you've got a cell wall that makes it much more difficult to divide.
Miss Ayers, you have been really disappointed. It was the other night at dinner. Blake said that animal cells have cell walls. I don't remember what we were talking about, but I remember he was like cell walls, and I'm like, it's an animal cell, and I was like, Miss Ayers would be so angry.
Pointed.
But that's like the basic, easiest thing to remember.
Doesn't he want to be a cardiologist? Oh, I failed him.
That's exactly.
How many classes has he had before?
That's kind of concerning.
I think the stress is getting to him.
We have to do this.
Oh crap!
Yeah.
I forgot about that completely. We need to do flashcards.
Yeah, we don't do those until tomorrow, thankfully.
Oh, because we're doing a role play today.
Yeah, today's the role.
Okay.
What? Tonight.
We're doing yes.
Riley can I are doing the role play.
Yeah.
All right, with cytokinesis, animal cells will pinch themselves in two. They form basically what's called a contractile ring. And the contractile ring, think of it as, okay, so have you ever seen those videos where they put a bunch of rubber bands around a watermelon?
Yeah.
Yes.
We had to watch that last week.
That's basically what's happening with the cell. They have a rubber band, essentially, around the cell that squeezes and squeezes and squeezes until it pops into two. So that's the contractile ring, is the rubber band that squeezes the cell and creates what's called a cleavage furrow, which is that indentation, until it pops into two. Now, obviously, it's not as dramatic as a watermelon because it's about to explode. But that's kind of what's happening. Is the rubber band is creating a cleavage furrow, and it eventually will snap the cell into two different cells. Bless you.
Like itchy. Paralyzed. No.
Do you know what?
Sure.
Is it like product buildup? It could be product buildup.
How do I get rid of that?
Double shampoo. Or shampoo twice.
I do shampoo.
I always, I go here and then I'll go back here.
No,
Really, your head can just be itchy, it's fine.
Yeah, I washed my hair last night, but I didn't use conditioner. That's what my mom said this morning, and I was like, I don't know. It saved the world to me, though.
There's you can go.
I was invested in your conversation.
So he's like,
Oh, it's product buildup. No, we just didn't.
which is product buildup.
All right, cleavage furrow is here's a cell that is a kind of a three-dimensional view of the, and then a cleavage furrow that is in between, caused by the contractile ring. Now, this is a plant, and the plant does not have a cleavage furrow. So, with an animal cell, basically what's happening with cytokinesis is it's dividing from the outside in. So this one, dividing from the outside in. With a plant, what we're going to see is that it's dividing from the inside out. It's making a cell plate in the center, and the cell plate is growing from the inside until it reaches the cell wall, and so it's dividing from the outside, from the inside out. So here we have our animal cell with our cleavage furrow. It's making this indentation, our cleavage furrow, and then eventually it's going to break into two cells. Because there is no cell wall present in an animal cell, it can just pinch in half like that. So again, same thing, cleavage furrow, snapping into two individual cells. Alright, so dividing plant cells face a different challenge because they have that cell wall. They have to form a cell plate. So basically what happens here is we have a bunch of vesicles. We all remember what vesicles come from, right? Where do we get vesicles? Golgi apparatus, Golgi body, forms vesicles, and inside these little vesicles or bubbles are the components of a new cell wall. So these vesicles are going to line up in the center, and the vesicles fuse together, and a new cell wall forms within this, which is called the cell plate. These vesicles will extend until they meet the existing cell wall, and then a new cell wall is formed in the center. And that kind of grows and separates the two, or into two cells. So the cell plate will form into two new cell walls. So one of them goes outside in, the other goes inside out.
I can't read it that well.
No.
No chips. No, I don't want these. Corey, do you want chips? Who wants chips?
I don't want anything salty.
I'll give them to you.
You don't like the chip?
No, but I packed them anyways. But I don't want to eat them. word in a sentence.
Okay.
That's what I did.
Okay.
You were in my dream class.
Oh.
Do tell.
Okay, so we were at my grandparents' house. I'll say the limit. But we were at her neighbor's house, which I have never been in. And you and I went in there, and there was like this giant Great Dane, like 2-year-old man. Like the house was just really messy. It was really
We were just in that person's house.
we just went in there.
Hey, was the door unlocked?
Or no, like the old man opened the door and like the Great Dane attacked us. Like got on our
I
shoulders.
remember when there was like this thing where like apparently your dreams like meant something. The people were telling.
I hope not.
My Great Dane was probably trying to attack you with kisses.
We like licked the side of
Well, there's like people telling me that like whatever you dreamed about was either something that happened or something that's gonna happen. And I was like, that makes no sense because I've had many I've had a nightmare when I was in like first grade
I
about
always have like the
Zootopia.
same 3 real
So I really hope that
like it's just a rotation.
I don't.
I just die.
I have different ones. I know it's about demons. Like a demon thing that hit my bed or something.
Mine is always like me having to like run away and like I get separated just like I get like caught and tortured.
Oh.
Like here's this one dude.
I think this thing
Every
every
secret,
secret.
I was like, I didn't know what to do. So I finally like, I eat the food. I escaped because I actually jumped out the right window last time.
Alright. So this is the cell plate formation. These are the vesicles. The vesicles are lining up by the spindle fibers. Spindle fibers have a lot of things to do. Lining up all those little vesicles, and inside the vesicles you have the parts for the cell, the new cell wall. So they're going to gradually fuse together, and as they're fusing together, they're forming the cell wall within them. So this is kind of forming the cell plate, and they'll continue to form and make that cell plate until they grow out and meet the existing cell wall of the old cell wall. So here you can see how this works. Here's our old cell wall within the plant cell, and like I said, the slides that we have are mostly going to be from a plant, and so you will be able to find the cytokinesis happening, and you will be able to see cell plate formation. You just might have to move the slides around a little bit in order to see this. So here is the reforming of the nucleus, which is happening during which phase? Does the nucleus reform? Coming back? Which phase is that? It's in your notes. Nucleus reforms and chromosomes unwind, decondense.
Oh, telephase.
Telophase. So this is telophase.
I was gonna say try.
I was getting there a little bit.
And also we have a cell plate that you can start to see forming right here in the center. So cytokinesis is taking place and telophase is taking place in this cell. Alright, entire process.
Here we go.
Actually, maybe just be cytokinesis, I can't remember. I can't remember which video. I know it is.
Cell division is required for an organism to
Entire process.
mature and maintain tissues. During the mitotic phase, a cell will undergo mitosis to form 2 new nuclei, and then divide to form 2 new individual cells during cytokinesis. Mitosis is the process of dividing the duplicated DNA of a cell into 2 new nuclei. Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears. Next comes prometaphase, where microtubules attach to the chromosomes. This step is followed by metaphase, where the chromosomes align. Metaphase is followed by anaphase, where the chromosomes separate. Finally, during telophase, nuclear membranes reappear around the 2 sets of chromosomes. Mitosis is now complete. After mitosis, 2 new cells are formed by a process called cytokinesis. Mitosis is only one part of what is called the cell cycle. For many eukaryotic cells, a cell is duplicated every 24 hours. Most of the life of a cell is spent in interphase. Interphase consists of 3 stages called G1, S, and G2. G1, or gap 1, is the first growth stage of interphase. In G1, the cell grows to nearly its full size and performs many of its specific biochemical functions that aid the organism. Next is the S, or synthesis phase. This is an important stage because it is during the S phase that DNA in the nucleus is replicated. The cell next enters another growth stage called G2, or gap 2. It is during G2 that the cell finishes growing. Once the cell has duplicated DNA in the nucleus and 2 centrosomes have appeared in the cytoplasm, mitosis can begin. For a typical eukaryotic cell, this will last about 80 minutes. During the first stage of mitosis, called prophase, we first see the classic chromosome structure. This occurs through a condensation process. At the same time, protein strands called microtubules appear from the centrosomes in animals. Finally, a structure found within the nucleus, the nucleolus, disappears. Next, prometaphase begins when the nuclear membrane is broken down. At the same time, microtubule strands, or spindle fibers, are growing from the centrosomes. These strands attach to a protein structure called the kinetochore. One kinetochore is attached to the centromere of each sister chromatid. Next comes metaphase. During this stage, the sister chromatids align along the center of the cell so that both chromatids face toward opposite poles of the cell. Now, the sister chromatids are ready to be separated. This occurs during anaphase through a shortening of the microtubules attached to the kinetochores. Additionally, the poles of the cell move farther apart, causing increased separation of sister chromatids. At the end of anaphase, the sister chromatids have moved to the 2 ends of the cell. Telophase is the final stage of mitosis. It is here the components of the new cells begin to appear. At this point, the spindle fibers are broken up. A new nuclear membrane surrounds the chromosomes at the end of each cell, and the chromosomes uncoil and return to an uncondensed state. Mitosis is now complete. The formation of 2 cells is all that remains. Following mitosis, the cell undergoes a process called cytokinesis. First, the cell is compressed by a contractile ring that divides the cell in nearly equal halves. By now, the organelles in the cell have been replicated and are now divided between the 2 halves of the cell. This includes mitochondria, Golgi bodies, and the rough ER. Plant cells also have chloroplasts. Once split, the 2 new cells are now fully in the G1 stage of interphase and ready again to begin their growth. Let's watch the process one more time. Mitosis begins with prophase. Notice the DNA condensing into chromosomes during this stage. Microtubules appear during prometaphase, and the nuclear membrane breaks down. Metaphase occurs when the chromosomes are aligned at the center of the cell. During anaphase, the chromosomes are moving apart. The telophase stage is marked by the appearance of new nuclear membranes. This is the end of mitosis. Finally, the splitting of the cell occurs during cytokinesis. The 2 new cells are now ready to grow and perform their specialized functions.
Cut. Did I just delete the whole thing? I think I did. What? Hold on, I gotta find it. Ugh.
I was on FaceTime.
Don't worry. It's right here. I think that was like close to the last slide though. Hold on. 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 Remember IPMAT-C, as I mentioned, interphase and cytokinesis are not part of mitosis. They are part of the cell cycle, not part of mitosis. Mitosis is a smooth, continuous process, so you're going to find stages that are in between. Spindle fibers and microtubules are part of this, like the cytoskeleton, and centrioles are only found in animal cells. So just some things to keep in mind. This is all in Google Classroom, so you don't have to write it down.
That's an awkward laugh.
And then that's it. So, new assignment. You ready?
No.
We're good.
No, yeah. What did you say? I have to read. Okay.
Have you started that?
Yeah, I started.
Fine. Where's your guys'?
Oh, yeah.
So I need
Ask
to-
you to do this. Can you
No,
make
not that one. Oh wait, I have to push pause on my video. That's important.
Yeah, because I'm going to cram in. I think I'm going to try to make my friends.